Welcome to the Neuroveda podcast for complex health. We feature expert guests who dive into ancient and cutting-edge understandings of health and disease. These conversations are about food, lifestyle, and medicine just as much as about nature, culture, and politics. I'm Jillian Ehrlich, family nurse practitioner, certified in Ayurveda and functional medicine, coming to you from our clinic, Neuroveda Health, in Seattle, Washington. And I'm actually really excited about our guest today. And you'll find out why kind of as we go along, because it's longer than I can say in just one sentence. His name is Thomas Mandel Clausen, and he completed his PhD in clinical cancer research at the University of Copenhagen and his postdoc at the University of California in San Diego, and has spent the bulk of his career thus far focused on cancer biology, malaria vaccine development, and more recently, COVID-19. His special focus was and remains cell and glycobiology, which will be the focus of our discussion today. Anyway, I'm going to not say I'm excited again. I'm just going to go forward. He's presented at conferences all over the world and has over 40 published articles within the field of glycobiology focused on various molecular structures and functions and how they relate to human disease. He's currently matriculated at the University of Hawaii to gain his medical doctorate degree with estimated completion in 2026. And what's fascinating about Thomas is he's actually a second-generation glycobiology researcher. So his dad was really into this. And so I can only imagine growing up at a dinner table, learning about this from the beginning. So Thomas, welcome. We're so happy to have you.
Thank you. Nice to see you.
Thank you. So I want to read something that just kind of is going to get us started, and then we'll kind of launch. So in preparation for this, of course, I did some reading and some prep. And so I just want to say the endothelial, actually, we can do like timeouts in this. So I'm going to actually fix this. Let's just start. We're going to start with kind of the biggest umbrella and then dive down. So tell us, what is an extracellular matrix?
Wow. So that's a very wide term. So an extracellular matrix is basically, so when you think back to school and you think to your biology class, they were teaching you a lot about cells. So cells is sort of a confined environment that is surrounded by a cell membrane and then everything contained within. The extracellular matrix could be a lot of things, but basically we use that term to refer to anything outside of the cell. So everything that holds the cells together, everything that holds together our tissues, and, because, within a tissue, cells within a liver versus a kidney have different functions, but they also need to be held together in a larger structure, which is largely done by the extracellular matrix. The extracellular matrix contains a lot of structural proteins, collagens and other things, as well as glycans. Actually, glycans is a quite big part of that. So a lot of functions in structural stability, in intercellular communication, all of those things are the extracellular matrix.
Extracellular matrix. Okay, so am I correct in understanding, like, when we think about the body, we think about it structured as cells. And so we think of like little globules of cells kind of like linked together. And I remember at one point, when you see even in high school biology, like a picture of the cell, it's like, there's a lot of space in there. There's like, they don't show it like it's real tightly packed. They show it like, here's some endoplasmic reticulum, and here's a nucleus, and here's a mitochondria. But the truth is that there's a ton of stuff in there, including like a whole like gelatinous matrix, and that outside of the cells, each cell kind of sits in this matrix as well. Is that right?
Yeah. I think the pictures we see in school are quite simplified. Obviously, a cell is not just a cell. We have many different types of cells. Otherwise, we couldn't be a complex human structure. And it is true, they show the endoplasmic reticulum, they show the Golgi, they show, mitochondria, and they might show one mitochondria within the cell, but in reality, a lot of cells have a lot of those, right? So it really, it's a simplified view. And I'll get into later as to why for glycobiology, that simplified view is a little bit of a problem, because actually the teaching of glycobiology is very different. So you can imagine a cell that has specialized a certain function within the human. It might be a liver cell, it might be a heart cell, a muscle cell or something along that line. It's specialized its internal structure to perform the function that it needs, but it also needs an outside scaffold and, in order to perform its function. And that's the extracellular matrix. I mean, it gets much more complicated than that, but that's a simplified view.
Yep. And this extracellular matrix comes, is my understanding, it comes evolutionarily from the mesoderm, like the other connective tissues. Is that right? Can you speak a little bit about the relationship between like extracellular matrix and connective tissue?
Well, I think in a simplified view, I mean, the extracellular matrix is the connective tissue, right? So you're right. It comes from mesoderm, but it also contains a lot of cells and components that are secreted by the cells and, all of these things. So but yes, connective tissue is what we would call it.
Okay. And I call attention to that just because the population that we see in clinic and a lot of folks who are listening to these podcasts often have connective tissue disorders, either congenital like EDS, Ehlers-Danlos syndrome, or acquired autoinflammatory or autoimmune. And so there's a lot of focus on connective tissue. And so it's interesting to think about, if we've got disrupted connective tissue, if extracellular matrix is also part of that, then the structures that can be disrupted with some of these connective tissue disorders might also be disrupted in the extracellular matrix.
Yeah, so now you mentioned Ehlers-Danlos, right, which is a collagen disorder, right? So that is specifically a disorder on the extracellular matrix and how it's built up, because there's a collagen defect, right? Other connective tissue disorders are the same, but it is a defect within that connective tissue, which is the extracellular matrix and other components of anything that is extracellular, if that makes sense. Yes, right?
Yes. So this is the part where I get really excited, because as we were talking a little bit before the show, my background in Ayurvedic medicine, in Ayurveda, we are much more space than we are structure. I feel like in Western medicine, we're often structure first, and then we're like a body that houses a spirit. Whereas in Ayurvedic medicine, it's almost the opposite. We are a spirit that is housed in a body. And the reason that that's relevant is because what I think we miss in Western medicine is how much communication, cellular communication there is, and is necessary for the whole kind of magical human being, like a single human being or a single animal of any kind, any species, really needs to function. So when we talk about the communication between the cells, and that's where the nervous system is, right? So it's the sensing, it's the processing, and then it's the behaving of the cells, the nucleus, the DNA, the RNA, the protein construction, the extracellular matrix, the inflammatory components, the fight or flight versus rest and digest of the nervous system. All of those things are based in perception and then processing and communication of the cells themselves. And it really seems like this extracellular matrix and this glycobiology is a lot of where that communication, where that perception, that sensing, that processing, and that behavior is sort of contained. And I'm curious your thought on that.
Well, I mean, to take it back to school cell biology also, right? In order for cells to communicate with each other, they need to be able to send a signal across the vast intracellular space, which is the extracellular matrix, right? And in order to respond, they need to have an intracellular, response cascade. So basically, a cell secretes something that needs to travel across the extracellular matrix or the intercellular space. It needs to bind something on a membrane, which is typically a receptor, right? And I remember my cell biology teacher back in college used to say, structure dictates function. So basically something binds, there's a structural change that transfers through the membrane, and now there is a chemical reaction on the other side that translates into a response by that receiving cell. Right. So you have, a complex cascade of a cell that chemically changes in order to release something, or biochemically changes in order to release something that travels, binds a distant cell. Now it gets biochemically translated and you get a response. Right. So the sort of simplified, if you focus on the cells, then you're focusing on the cell's response to a soluble factor and how it responds to it. Mm-hmm. But you also really have to look at the extracellular matrix, because that signal is sent across that extracellular matrix. I mean, also blood and other things, but like the intracellular space, right? And how signals translate across there, that's an interesting question, right? And that's where glycobiology kind of comes in, as you said yourself. So I did a lot of work in glycobiology and cancer with my colleagues at the University of Copenhagen. And one of the things that cancers do is that they make a very dense, it's not like glycocalyx, but they put a lot of glycans within that extracellular matrix. And what they do is they kind of function as a sink for certain growth factors and cytokines and different things that kind of creates a local environment of very strong signaling that drives cancer growth, right? So that's one way the extracellular matrix sort of adds to, cancer's growth. And it's an example of how changes within the extracellular matrix can change intracellular signaling, if that makes sense. Yeah. So I think it's a very important thing it's a very complex topic that we don't really fully understand, obviously, but that's an example.
Yeah. Yeah, it is. Yes, it is very complicated. So the mechanism that you're describing, though, we can kind of all understand in terms of like there's signals that are sent out, they have to cross this matrix, then they're received. And, that is a process that on, somewhat of a human level, we can understand, making a signal, passing a signal across a space, receiving a signal. And that making conformational changes, which is what we would call it in the cell, but making behavioral changes is what we would say on this kind of like species plane on the level of people. So I'm wondering if kind of with this understanding that this is the world we're working in, you can make some definitions for us. So like, how would you define glycobiology? What does that mean?
Well, well, I mean, very basic. It's the study of sugars, but using the word sugars is a little problematic because people think of the sugar they put in their coffee or in their tea, right? Or in chocolate or whatever, and that's not really the case. I mean, it is the study of carbohydrates, and carbohydrates can be many, many different things. There's many, many structures. They have the same basic structure, which is why we call it carbohydrate, but they can take many forms. So glycobiology is basically the study of carbohydrates and how they affect, biochemical reactions within the human or within, doesn't necessarily, it could be animals, it could be plants, it could be anything, but it is just the study of carbohydrates and how they function biochemically. Does that make sense?
Yep, absolutely. And so how would you define like the word glycan?
But glycan is just kind of the shorthand we use for carbohydrates.
Okay, so those would be interchangeable. So a glycoprotein, what would that be?
Glycoprotein would be a protein that has, a carbohydrate or glycan on it, which my father would probably say all extracellular proteins do.
Okay. And then how would you describe there's a word glycosaminoglycan. What would that be?
That's a very specific type of carbohydrate, which, I mean, we could spend hours talking about the different types, but glycosaminoglycan is one, and it's the one that I have studied the most. Glycosaminoglycan, it's these very, very long carbohydrate chains of disaccharides that are repeated and then modified. So they make up a very big portion of the extracellular glycocalyx as well as the extracellular matrix. And they have a lot of functions in disease processes, which is what my career has been mainly focused on. So glucosaminoglycan, you might know some of them. So obviously hyaluronic acid, heparin sulfate, which is, heparin is a type of heparin sulfate. But heparin is not heparin sulfate. It gets a little complicated, but and then chondroitin sulfates. Those are your main glycosaminoglycans.
Okay, got it. And so heparin is an anticoagulant. We make it internally, but we also use that in the hospital if you're trying to anticoagulate a patient. For example, somebody comes in atrial fibrillation and you don't want them to have a stroke, and so you might give them heparin acutely to anticoagulate the blood. And then we also make heparin endogenously in our vessels, correct?
Yeah, so heparin is, everyone has heard about heparin, at least within clinical setting, but I don't think they fully understand that it's a glucosaminoglycan. But it is probably the most used anticoagulant, and it is a sugar. What it does is it binds a protein called antithrombin 3 and activates it, thereby inhibiting quite a few points within the coagulation cascade, primarily thrombin. So that's how it exerts its role. And we do, it is produced in our own body as well, but it's thought to be kind of a rare molecule in humans, which is why we can give it exogenously as a treatment.
Got it. And hyaluronic acid, I mean, we use that. I mean, that's in a lot of beauty products now.
It is, and that is because the unique properties of these sugars, I say sugars, carbohydrates, they bind a lot of water. So hyaluronic acid, is able to bind a lot of water just by the chemical properties of the sugar. So it generates this gel-like thing, which is good for hydration on the skin. I mean, we know that it's good in joints for good joint health. We know it's good for many things. But hyaluronic acid, other than being in beauty products, is actually a very big part of our cellular glycocalyx and replication, all these different things on cell surfaces.
Then chondroitin, chondroitin and glucosamine, which is another one, like, those are what we give often, you can buy at Costco, like for arthritis, or you can like give to your dog when they get older.
It's funny, I was in clinic the other day and we had a discussion about chondroitin, and, there was a lot of evidence suggesting that it would work, and then some that says it won't work. I mean, there's no doubt that chondroitin has a very important function endogenously for joint health. The question is whether or not eating it can deliver it to the right location, if you understand what I mean.
Yep. And we talk a lot about that with collagen as well. People eat a lot of collagen in order to try to build collagen, but I'm curious your perspective if taking collagen orally, like in these protein shakes, can actually deliver collagen to joints.
I mean, I have to be careful what I say. It's a non-twist, but I would be skeptical because, proteins that enter your GI tract get degraded, right? So mostly you'd be eating amino acids, which could then be made in collagen.
And the problem of arthritis is not necessarily a lack of nutritional components. It can be, anything can be a lack of nutritional, but often it's either too much inflammation or it's a quicker breakdown. So that the problem of the arthritis is inflammation versus a nutritional deficiency.
Yeah.
So we've talked about some of these types of glycans. What is a glycocalyx?
Calyx is the word we use for the carbohydrate covering of cells. That's the simple way to say it. So again, if we go back to that cell biology view from your college biology class or high school biology class, they show a cell membrane, which is lipids, and then they show a couple of proteins within that lipid layer, some of them that bind on the outside, some of them that transverse through the lipid layer in order to transfer signals across, right, or different things across if it's a channel. So what they show is typically a lipid layer, some protein that sticks a little bit off the lipid layer, and then they might plug a glycan in there to just show that, oh, there's a sugar here, but it doesn't really show that in reality, what you have is a very, very thick layer of glycan that basically covers all cells. So it's, the simplified view makes it look like anything can just kind of diffuse into the cell membrane, bind to a protein, cause some kind of signaling within the cell, which is not really the case because anything that needs to get to the proteins in the cell membrane really have to transverse through a very, very thick layer of glycans, which I've always thought of as being sort of when we were working on COVID, someone asked me how to describe it. And I described it as a very thick forest. And, the bird that needs to get through the forest in order to get to whatever it needs down on the grass. Does that make sense? Right. So that's the glycocalyx. The glycocalyx contains a lot of components. It's glycosaminoglycans, it's mucins, it's all types of glycans that we could talk a long time about the different types. But what it does is it generates a barrier because it binds a lot of water. It also, a lot of these glycans have functions in binding certain soluble molecules that we just talked about. So it has a big impact on cellular signaling, structural component to it. There's a lot of functions to the glycocalyx.
It's fascinating. Sorry, a lot of questions. So it's almost like, I like that forest analogy. And, there's tons of movies that have like the magic forest where like the trees are alive. And, there's some little boy walking through trying to find, and then in this kind of like magic forest where it can be the weather can be something above or something, something below, but then in the forest, everything is a little bit kind of on its own. I'm curious, what directs the glycocalyx? Like how does the glycocalyx essentially, like, I don't know how else to say it, but like make decisions about, how does the body make decisions about what the glycocalyx is composed of? Like which proteins are there? Where do they move? How do they function? What directs that?
So that is a subject of intense research. We do know a lot about the glycocalyx, but for example, how it's made by the cell, right? So it's made through the the Golgi, and then put on the cell membrane. Some of it is excreted into the extracellular matrix, and some of it stays on the cell membrane, bound to protein and other things. So I can mostly speak on glycosaminoglycans, which is the toughest glycan to research because it's a non-template driven production, right? So our cells produce it. When our cells produce DNA, there's a template. So we know that when a cell starts replicating its DNA, we know what it's going to make, if that makes sense. right?
Mhm.
With glycosaminoglycans as an example, there is no template. So exactly what the cell produces and how it varies the structure of what it produces is a little bit something we still need to figure out fully, if that makes sense. Now, there are other glycans that are a lot more predictable, but exactly why a cell puts certain things on the surface is not something that is fully understood.
Fascinating. And glucosamine is big, is it not? It's a big.
Yeah, glycosaminoglycans are big, yes.
And so is it kind of like, could you imagine like, frost moving across a window in terms of like, where does the frost particles go and how do they look? It's kind of like, it's somewhat like there's a pattern, but then it becomes somewhat environmentally driven.
Well, so the cell produces it and puts it on the cell membrane, right? So the way I've always thought of it is, so a proteoglycan, which is a protein that carries a glycosaminoglycan, is produced and matured within the Golgi, and the sugars are made, and they're these long sugars. And then the protein is put on the cell membrane, and what you have is a protein with a long sugar that kind of extends into the extracellular space, right? So it's not being produced in the environment. It's being produced intracellular and then put outside.
You frost the window and then put it in the hole of the wall. Fascinating. And just to go back to sugars, just some very basic, because I realize we should just cover this. There's, talk to us about like a monosaccharide, a disaccharide, and polysaccharides, because a lot of folks who listen will probably know about like the FODMAP diet. Do you know FODMAP diet with disaccharides? Okay.
They're not.
Fructo-oligo disaccharides. And then, so that's the diet, like if you have SIBO or small intestinal bacterial overgrowth, it's one of the things that they'll recommend is a FODMAP diet to avoid those disaccharides. But can you talk a little bit about like what's a mono, a di, and a polysaccharide?
Well, so it just the chain length, if you will, right? So monosaccharide is either, for example, hexose or pentose. That is just the basic unit of the sugars we're working with. That would be the monosaccharide. Disaccharide is two saccharides put together. Mm-hmm. So that would be your fructose, your, things that you get in through your diet. Now, carbohydrates that are longer are typically made up of repeated or like structured monosaccharides or disaccharides. In the terms of glycosaminoglycans, is really a disaccharide that is repeated anywhere from, tens of times to hundreds of times, making these long sugars. Now, sugars, your intestines have to break them down to manageable fragments in order to pull them up. And that's what your GI is doing through all these enzymes, right? It's breaking down first complex carbohydrates into more manageable pieces, and then it's breaking it even further down, disaccharides, monosaccharides, in order to be able to take it up into the body, if you understand what I mean. Which is kind of we were talking about eating complex carbohydrates. I mean, it is, we don't take up complex carbohydrate, we take it up as building.
And the complexity of it often means that there's other elements with it, or that it digests a little bit more slowly versus something like just eating, table sugar, just eating glucose, or just eating like glucose gels like they give for marathon running, or in our environment, we're used to eating more complex foods, and having the process of breaking them down is what we think of as for health, because that's evolutionarily how we evolved, correct?
Exactly.
Okay, so these, in my little prep for this, I was learning about how one of the first articles on the glycocalyx came out in 1970, called "The Surface Coats of Animal Cells," and so, or thinking about it as a sugar coat, that gave me a kind of a clear picture of that around these cells. And it's not just around each cell, it's also in like the endothelial linings of the blood vessels, correct? I mean, I guess those are cells, but that would be an endothelial glycocalyx. Yes?
But that is still cells. It's the endothelial cells that are covered.
Okay, got it. Okay, so we have this like magic forest. I call it that forever. Thank you for that. So this glycocalyx, there's essentially all of this biology that's happening that's kind of driven by the cell that now exists kind of around the cells. How does that impact disease? Like so first of all, let's actually pause and say, what are some of the functions of the glycocalyx? Like what are they doing?
So that's a vast, it depends on what you're talking about. So if we talk about the endothelial glycocalyx, right? So the endothelium lines all of our blood vessels, right? and sort of makes up the luminal lining of a blood vessel, right? So what is the blood vessel doing? Well, it's carrying, obviously, the cells of the blood, red blood cells, the immune system cells, as well as all types of soluble factors that, cells use for communication, and as well as nutrients and all other things, right? So the glycocalyx is sort of the first barrier, right? For anything that needs to get in and out of that blood vessel. Yeah. Because it is the immediate component, or the component closest to the lumen, right? So I've always thought of the endothelial glycocalyx as primarily being lubrication is one, it binds a lot of water. There are some quite cool experiments done a long time ago that kind of shows that within a blood vessel with flow of red blood cells, they actually kind of line up within the center of the the blood vessel. And that is partly due to this glycocalyx layer just kind of creating a lubricating layer, allowing them to uninhibited flow through within the center of the blood vessel, right? So that's lubrication. We also know that the sugars have a lot of functional components to it. So one of them, for example, is this heparin structure, which is an anticoagulant, which is something that you obviously want there in case coagulation goes awry. The other thing that I always thought of it as is that it is a physical barrier. So you have a lot of immune system cells traveling through the blood as well. And, we all know what happens when the immune system goes too far. You get autoimmune disease, right? So it is a regulating factor as well. And we actually do know that if there is inflammation in a given place, what the cells do is they let go of that glycocalyx, allowing the immune cells to, exit the blood system and enter the tissue. It also, our blood vessels are very reliant on being able to sense changes to flow. So you could imagine having these trees that kind of extend into the blood vessel lumen, and the wind that pulls the trees. So that pulling is a way of sensing flow within the vessel. So auto-regulation of flow is what you would be thinking of there. So that's one of the functions of the glycocalyx. And then also just binding a ton of factors that needs to get in and out, or, for cell signaling purposes, cytokines, chemokines, growth factors, all these different things. So the glycocalyx has a lot of functions. I only touched on a few of them. I'm sure my colleagues would suggest that I picked up on all of their favorite as well.
Yes, I'm sure this is good. This is good, like late night discussion about like what is going on in there and everything that it's doing. So kind of in summary, things you talked about are like the selective vascular permeability, the modulation of coagulation. So certainly if you cut into your, if you have a cut, then you need those blood vessels to form a clot to stop bleeding. And if there's too much inflammation, you need that, or there's shear stress, you need that heparin there, or that antithrombin in order to keep that flow moving. So thrombomodulation, immunomodulation, all of the different immune factors, the mechanical signal transduction, and the localization and regulation of soluble factors, what moves in and out. Okay, so we have functions. What happens when those, what damages the glycocalyx, endothelial glycocalyx?
A lot of things too. Infection, inflammation, mechanical disruption. You could imagine many things that would damage the glycocalyx. Obviously, I'm a glycobiologist by trade, so I would say that damage to the glycocalyx is a hallmark of quite a lot of different disease processes. And what happens? Well, so we know that, for example, inflammation, there's some different thoughts on that, but inflammation largely leads to digestion and release of the glycocalyx within the inflamed area, right? So what does that do? Well, it leaves a exposed endothelium. Now we're talking about the endothelial glycocalyx. Obviously, there's also glycans within the extracellular matrix and other things that have different functions. But in the endothelial glycocalyx, if you have inflammation, you now expose a sort of a bare endothelium, which is now, immune cells are going to be able to invade that area. It's potentially going to be prothrombotic, that surface. So damage to the glycocalyx would cause increased inflammation, turbulent flow, potentially thrombosis. So basically not a very good thing to have.
And can you think of any? somewhat of a rhetorical question, but can you think of any disease or condition that would not involve the glycocalyx?
That's a good question. I mean, potentially anything intracellular, right? But even that could have effects on the glycocalyx. I think that the more I learn, and I think that the beauty about being in glycobiology is that it is one of those fields that is always evolving. I think I told you before we started, my father is a glycobiologist, my mom as well. But my father once told me that there is no biological interaction that doesn't include a glycan. So I know that if you asked him that question, he would say that, probably we'd be able to come up with a thing where we don't know there's a glycan, but most likely if you research it a lot, somehow glycan is involved.
I mean, you would never have, in my mind, you would never have any one disease process that would be like visible or experienced on the organism level that would occur like inside a cell that would not translate to anywhere outside of a cell. And so it sounds like from what you're saying that if there's any condition that occurs inside a cell that doesn't disrupt essentially glycocalyx production that's happening inside a cell. So assuming that that's not disrupted, there would never be a condition in which it would happen inside a cell and would not transverse the cell and reach the level of the organism that would not involve the glycocalyx.
No, because somehow it has to reach the level of the organism. Otherwise, you're not going to be...
You won't be sick.
Right? Yeah, exactly. I mean, I think it, at least in terms of research, it really comes down to what is the cause of a certain illness and what are the downstream effects, right? So I think you could probably find many causes that are not glycan related, but I think somewhere within it there's going to be impact on the glycosylation within the diseased area, if that makes sense.
Yeah. And so how much of the glycocalyx is locally versus kind of system-wide controlled? Like, so for example, you can have inflammation that's a local, or you can have kind of a systemic inflammation. And I'm curious, like, changes in the glycocalyx due to inflammation or due to disease, how much of it is local? Like, clearly, if I crush my finger in the door, that will be local. But how much of it is also, can you affect a glycocalyx systemically, like the whole thing at once?
That's a good question. I mean, that's also a question that would need some research. I think it depends on. So if you locally hurt yourself, there's going to be a lot of local signals saying something went wrong here, right? The immune system is going to respond locally. There's going to be a lot of cytokines and different things saying here is something we need to respond to locally, right? Definitely the glycans will be affected locally. But when you talk about systemic disease, then the body is responding systemically, right? So for example, your immune system is going to respond, which I think is what you're referring to, right? It's going to respond systemically. And we do know that, and now I'm going to talk about cytokines as well, because it's one of the signaling molecules that people know, but that they can have effects on glycan production and how the biodynamics of the glycocalyx is performed, right? So in that case, I would say that there would also be a systemic effect on glycocalyx. Does that make sense?
But it sounds like we don't quite know enough about to really describe what that mechanism, like how that mechanism unfolds. Is that?
I mean, we do know some things, but I mean, it is still an area of intense investigation. So how the dynamics of glycocalyx changes in different disease states.
Let's talk about a few of those disease states. And I'm sure nearly foremost on everybody's mind after I read your bio is kind of like, what do you know about?
Well, so COVID was kind of an interesting experience for me and the group I was working with. I mean, I think if I could take a step back and say how we got into it. So I was a researcher in Copenhagen, and I was given an opportunity to go work with an expert within the glyco field, Jeffrey Esco, at the University of California in San Diego. He invited me into this lab, and we were going to work on cancer, essentially. And we did so for the first year I was there, and then COVID hit. And I think the entire medical and scientific field suddenly shifted their focus to COVID. Now, the group I was working with in San Diego, they are experts on heparin sulfates. Probably they were one of the world's most leading groups within heparin sulfates. And there was this report suggesting that COVID-19 infected cells through heparin sulfate. It was just a minor note online, and everyone was sharing. Me and a couple of my colleagues thought, well, I mean, we're in the lab that knows heparan sulfate, so why don't we look into this? And we did. And we spent, as a group, a couple of months really intensely working on finding out whether or not heparan sulfate was involved in cellular infection of the SARS-CoV-2 virus. And it was. We published that if you remove heparan sulfate, which is a type of glycosaminoglycan, from the cell surface, you do not get COVID infection. The mechanism was, again, this tree, right? So you imagine a virus, it needs to come in and bind a cell. It needs to get through the glycocalyx in order to get to the cell membrane where we know ACE2 receptor is, right? And then it needs to get through to get inside in order to replicate, right? That's what a virus does. So if you are a smart virus and you need to get through a very thick layer of glycans, well, then you evolve to interact with it, right? Not to guide yourself through it. And that is basically what we showed, was that the spike protein binds heparan sulfate. That actually primes the spike protein to kind of expose its binding sites for its protein receptor on the cell membrane. And if you don't get that interaction, you don't get infection. That was what we showed. So it's kind of a side project, but also a very, very, very interesting project that meant a lot to my career and to the group in San Diego.
What can we do with that? Like, what does that teach us, or what can we do with that?
Well, I mean, there's a lot of ideas then, right? Because that teaches us. So basically now we know something about the mechanism of infection for the COVID virus. Well, basically, when we're developing medicines or anything that targets infection, because we want to prevent virus from infecting us, right? We look at the mechanism to see, well, where can we intervene? Now we know that the binding to heparan sulfate is one way of it. So for example, we could make an inhibitor. I mean, it could be heparin that binds the spike protein and neutralizes the virus, right? Because the spike protein wants to bind that structure. Heparin is probably not, I'm not suggesting that heparin is treatment, but like something along those lines, right? Or you could imagine making something that changes the glycosylation within the cell, making the cell resistant to infection. There's a lot of things. I mean, and COVID is just one example of this. There's multiple viruses that use glycans for infection. I mean, probably almost all of them, right? So I mean, I guess the most well-studied is that we know the flu viruses use sialic acids to get into cells. It's not a new mechanism. And actually, if you look through the literature, you will see that the viruses that came before SARS-CoV-2 also used heparan sulfate, brilliant SARS. So that was 6 months of work in 30 seconds.
Great. And so what else do we know about kind of the impact of COVID? And I'm wondering if you can speak at all to what we think might be going on with the endothelial glycocalyx and long COVID or chronic inflammation.
Yeah, so that is something that we've talked about a lot, and something that a number of my colleagues are interested in, and that I've personally been interested in. So right now, the data is a little fuzzy. There's it's not fully understood what's going on. But what we do know is that just the presence of the spike protein or the inflammation that comes with COVID infection leads to shedding, which is digestion and release of the glycocalyx from the endothelium and other cells, infected cells, right? So there are some research out there showing that that long COVID patients have COVID components circulating in the blood, such as spike protein, from that previous infection. And there are also papers out there showing that fragments of the endothelial glycocalyx are circulating in the blood months after primary COVID infection. So the idea there, and I mean, there is, I have to say, there's data published that shows both ways, but the idea there would be that the COVID patient, the long COVID patient, is in some state of sustained dysfunction. It could be, inflammatory. It could be many different things. There are many hypotheses out there. What we specifically have been interested in is the health of the endothelial glycocalyx, because if a patient has a sustained insult to that glycocalyx that leads to glycocalyx dysfunction, that could explain a lot of the things that people are seeing, neurocognitive problems, which relate to trouble flow through the brain and things like that. So that was my hypothesis, well, not mine, but the hypothesis within my field, if that makes sense.
And so how would you Are there ways how do you repair damaged endothelial glycocalyx?
Well, if you remove the insult, the cells will do it themselves. So, and it actually, the cells are very good at making glycans. They recycle them all the time. So the real question is how to speed that up or how to prevent that it gets degraded. Not degraded. Degradation. Sorry, is through a number of enzymes. It can be proteases as well as enzymes that specifically degrade the glycans within the glycocalyx. So you could imagine making inhibitors of those enzymes, but basically protecting the glycocalyx from the beginning so that it doesn't get degraded. Other ways of doing it would be to try and find out how you can induce cells to produce glycans faster so that they can regenerate faster if there had been a problem. And both of those strategies are things that people are working with.
And so I'm curious, in your opinion, there's kind of camps about like persistent viral presence versus, essentially parts of the virus or an inflammatory cascade that doesn't include a persistent viral infection for in-long COVID. And I'm curious where you kind of sit with what you know right now.
So it is a complicated subject to read up on because, as you say, there's a lot of opinions out there. So what I know from the glycocalyx is that all those three mechanisms that you just described could cause endothelial dysfunction. So I'm more thinking that there is some kind of continuous insult that is causing problems, whether it's a sustained infection, retained viral products, or another mechanism. I mean, I've seen good arguments for all of those. So it's hard to figure out what's going on, but what I do think is that there is some kind of sustained insult that causes problems within the glycocalyx.
And are you familiar with Bob Navio at University of San Diego? Do you know that guy? Okay. All right. So we won't chat about him. He has a whole cell danger response involving kind of mitochondrial transitioning to be cytokine driven as opposed to ATP driven. But we won't talk about that. And there are, in the kind of integrative medicine world or the alternative medicine world, there are some like supplements that they talk about for endothelial repair. And I'm curious if you are familiar with any like supplements or nutrients or anything that can kind of help with endothelial repair.
I mean, I could imagine that there would be a lot of ideas out there. I have not seen anything that I would recommend as being a definitive supplement for that. But I mean, when you're talking to a basic biochemist, I've not seen any definitive proof of something that works that way. I know there's a lot of research into different components, but I've not seen any proof that would suggest that there is a good supplement out there.
And I'm curious with this COVID piece, like if we're thinking that it's impacting endothelial glycocalyx, can you talk a little bit about the similarities or differences between the extracellular matrix or endothelial glycocalyx we've talked about thus far and the blood-brain barrier?
So the blood-brain barrier is an endothelium, and it's with other components as well, right? So that is largely made up of how the cells are organized and how porous that intracellular interaction and through cellular interaction is, right? But the glycocalyx is also a big part of that. So exactly how it differs from glycocalyx in the periphery is something that is also intensely being investigated. Exactly how that works. There's no doubt in my mind that if you disrupt the glycocalyx within the blood-brain barrier, you're going to have defects in the blood-brain barrier. Answer your questions.
What was I had another. What was I going to say next? I mean, it's striking to me how much like the questions that we're asking kind of in clinic and the questions that we're asking for these patients who are really struggling or suffering with long COVID are like, we are on this like cutting edge of this. These are the questions to be asking. Oh, I know what I was going to say. So in some of the worlds that I've kind of been involved with over the last 15 years I've been doing this, they talk about this chronic inflammatory response syndrome. And there's this emphasis on VEGF. And I'm curious if you can talk a little bit about vascular endothelial growth factor, VEGF, and kind of where you think it might play a role in the pathology that we're talking about.
Well, VEGF, traditional, I mean, we know that it's important for vascular growth, right? Tumor cells use it. We use it, obviously our healthy cells use it in order to build new blood vessels and maintain health of the blood vessel. It is funny you mentioned VEGF because it does bind glycans. So, it would adhere to the glycocalyx. In terms of, I'm not too familiar with how it works in terms of chronic inflammation. But VEGF is a very important factor in multiple disease processes, and an important factor when we talk about glycans, because it likes to bind them and needs binding in order to work.
And what are some of the disease processes that VEGF is involved in?
One thing is cancer. Cancers rely on angiogenesis, and VEGF is one thing that could cause that. We also know that VEGF is a problem in neovascularization in certain diseases such as in the eye and diabetics and things like that. But I think it's, VEGF is just one example of a factor that our body obviously uses in health in order to provide a function, which misregulation can cause a disease process, right?
Mm-hmm.
Being located in a lot of if that makes sense.
Yep.
I think the main thing, especially when you talk about biochemistry, is that the main word is balance. Too much, you get problems. Just the right amount, you're healthy.
Yep, exactly. We're all looking for that sweet spot. Yes, absolutely. And so this is kind of fun. Usually I try to direct a show kind of like along a pathway, but now I feel like I'm just like lobbing questions at you. So thank you for being such a good sport about this. So few others, one thing I'm thinking about this magical living forest around our cells. And I'm curious, are there other organisms in there? Like, we know that the microbiome is so active all over the body. And I'm curious if you're familiar, we know that viruses kind of, once they're in your body, they don't necessarily leave. They just kind of hide in different cells or different places. I'm curious, what else is in that dark forest around the cells? Are there organisms in there? Are there biofilms associated with the glycocalyx?
I think it depends on where in the body you're talking. Within the GI, definitely the glycocalyx have a very important function in maintaining the microbiome that makes up the gastrointestinal tract, right? This is a question you should have said to my dad, because my father wouldn't know what to say about that. But so the glycans, both can serve as a point of food for a lot of these organisms, but also as a barrier keeping the microbiome where we want it to be and not in the blood where it causes problems. Does that make sense? Within the body, at least the traditional thought is that those types of organisms shouldn't be inside. It should be in the GI. It should be on mucosal surfaces, but it should not be inside where we cause problems. I know that there are certain studies that show that maybe bacteria do make it inside, and that's physiological, but it's still a debated topic, if that makes sense. So on an endothelial glycocalyx, you wouldn't find organisms, at least by traditional thought.
Okay, interesting.
But not unless there's a disease.
Yeah, not unless, right. Because there's definitely, there's definitely things that make it inside, but that would be pathologic versus physiologic.
Yeah, and that is, glycans make up a big part of that barrier to infection. That I don't call it an oxymoron, but the thing is that it makes up the barrier to infection, but also in many cases becomes part of the infectious process. And that is pathogens from the outside through evolution figuring out how can I manipulate this barrier and get through, right? So talking about COVID using heparin sulfate, you could talk about anything from Lyme disease to malaria. They all use glycans, which is an adaptation to use our own defense, right?
Yeah, fascinating. So, okay. So any thoughts about how we can essentially keep focus on maintaining or keeping or nourishing a healthy glycocalyx? How do we take care of our glycocalyx? How do we nurture those little forests around every cell?
I think you take care of it the way you take care of the rest of your body. And as a second-year medical student, I have to say diet and exercise.
Sleep, trauma recovery. Yes.
Preventing inflammatory processes. The more I dive into, medicine, because obviously I did biochemistry before and now made the crazy choice of going back to school and becoming a doctor. Well, diet and exercise. Yeah.
Right. And are there any ways to how we could kind of, even in conventional medicine, measure the health of our glycocalyx? Are there biomarkers that reflect?
Well, I mean, we're talking about, there's a lot of research into biomarkers for different disease processes, right? So some of the things that I've been involved in is, for example, we know that cancers overexpress these glycans. And I mean, we could spend hours talking about what that does to help cancer. But definitely in a cancerous state, we have a lot of evidence suggesting that the glycans that circulate in the blood change and change levels. So we know that by taking a blood sample from a patient, if we know what glycan or, either whether it is a glycoprotein or whatever we are, if we know what we're looking for, we can actually detect that there is a change to what circulates in the blood in a cancerous state. Now, we also have a lot of information showing that in a infectious state such as COVID, the glycans change. So you would imagine that you could use that as a biomarker for disease, which is something that I personally hope we could do for long COVID, for example, find markers through disruptive glycocalyx. So yeah, there's, and then so that's the diagnostic aspect of it. Then comes the whole therapeutic. How can we intervene,
Yeah.
So there's a lot to be done out there. I think, and one of the reasons that I've always liked glycobiology is that it is such an evolving field that people in the past, we're human. If it's hard, we try to avoid it, right? The glycans are hard because it's not as easy to define as a protein or a piece of DNA. It is, and for a long time we've been developing ways to investigate glycans, and it's been an intense area of study, but we have been limited in our way of characterizing them, specifically glycosaminoglycans. But we're coming after that now. And I personally feel that it's becoming more and more of an area that people now pay attention to compared to before, where the protein guys would say, oh, there's a glycan here, and then just keep going. Not really worry about it.
And my understanding, is this correct that, when we think about DNA, so first of all, when you think about the field of genomics, or you think about the field of genetics, you think about DNA making RNA. Okay, we have 20,000 genes, there's umpteen, million combinations of these genes. There's de novo changes, mutations with every generation. But in some ways there is a cookie cutter aspect to DNA, and that it is fairly patterned versus what you're talking about, which is even less regimented than DNA. So these glycans are not actually directed by DNA. They're not cookie cutter from DNA patterns. Is that right?
Well, to some extent they are, because the enzymes that make the glycans are obviously directed by DNA. Okay. But it's a funny thing, it's a funny concept now that you bring it up, right? Because for a long time, we couldn't explain how do you get an organism as complex as a human out of some 20,000 genes, right? It doesn't really make sense, right? So then we started talking about RNA being modified so that you can actually get more than 20,000 proteins out of a given DNA, right? Because a combination of the mRNA and all this stuff that makes more proteins. So then we knew that we could make more types of proteins, but then came the secondary modification, which glycans is one of them, right? So in reality, through glycosylation and other secondary modifications of proteins, we can make millions of different functional, right? So, and the problem is that, at least in the glycans, is that it's not template driven. So how does that work, right? So I think genetics is one thing. You can find a given mutation and this enzyme is going to be problematic and it's going to cause disease. We know that if you change RNA processing, you could get disease. But what happens if you change the secondary modification? Right now, you have almost an infinite amount of problems. Potential problems that could go on, right? We don't fully understand that. We do understand a lot. I have to say that, although my colleagues will be angry, but we don't fully understand it. So where do we go from here, right?
I mean, we could look at it. We could flip it also to say that, we have these genes that are somewhat protective for making this complex an organism. And then we have the mRNA, the messenger RNA, which could be more protective as opposed to seeing it as problems. And then we could say, we have all these millions of glycan structures now, which can also respond to the environment in terms of keeping us created as a person, keeping us with our structure and our function so that our pinky finger is always the smallest. I mean, some of those things are so simple yet so remarkable. The fact that like we all have, like, how many times do you see a pinky finger that's longer than all the other fingers? That almost never happens. Like we are really, all of our variety, we really are fairly kind of contained structurally in some ways.
Well, this definitely is a point of regulation for everything. The
Yeah.
question is, how is it done?
How's it done? Yeah. So that's where Ayurveda says, right? We're conscious, that's consciousness. And Ayurveda would say every cell is a center of consciousness. Every cell has mind and some component of mind. And I'm constantly trying to inject that into science because it doesn't really exist. We are machines in Western medicine in a lot of ways, but we are so complex. And I think this just really emphasizes that complexity. So we're ending on diet and exercise.
But I mean, there's other, I'm sure, other ways. You were asking about supplements. I don't know of any. I mean, we know chondroitin, people were taking it for health of their joints. Doesn't seem to be working all that great. So yeah, diet and exercise, stay healthy.
I don't know how much sleep you get as a medical student, but sleep.
Yeah, because I know sleep is important. So I do get my sleep.
Good. Any last thoughts before we close? Anything else you'd kind of like to share that you're like, places where the research is really exciting to you, or things we should kind of look for coming down the pike regarding glycobiology?
Well, so I think, so I started working with malaria and pregnancy, which is again a place where a parasite uses a sugar to maintain an infection within the human host, in this case, the placenta. I went on to work on cancer and how the glycans change in the cancer and how that helps the cancer do the bad things that the cancer ends up doing, unfortunately. That led into COVID, and how COVID virus and other viruses uses the glycans to get into cells. And the more I think of it, more I've been getting close to that endothelial glycocalyx, and what that does. And I think my enthusiasm is understanding how to be able to manipulate that glycocalyx benefit patients. And that's what I'm very passionate about and what I hope to, make some difference at some point. So
I love that. Good. It's tempting for me always in situations like this to launch into more discussion about Ayurvedic medicine, because I do feel like the ideas behind Ayurvedic medicine are, how do we shift our whole system understanding, or how do we shift, like, what I mean by our system is like our physiologic system. How do we impact multiple places at one time, as opposed to giving one supplement or giving one herb? There's a lot of synergy in Ayurveda, which does have a whole science of the daily routine and then seasonal changes and lifespan changes. But a lot of those treatments are intended to change kind of how we feel in our body and what our physiology is able to do, which goes back to what we're talking about, not close your finger in the door, but when you're trying to impact your glycocalyx all over your body, or all over your body, be it, there's one type of all over your body that's like endothelial glycocalyx, where it's all the vessel lining. So yes, we have vessels, we have lymph vessels, or we have nervous system cells all over our body. But then we have, the individual connective tissue that's the coating of every cell, and that determines all the different pockets and areas, our peritoneum and kind of the sacs, the bags of cells that, if you had a little hole in your head, you could just kind of drop in all those bags of cells. So it'll be really interesting. And maybe in next year we could do another follow-up to this, kind of what you figured out, what you've learned, and where we're at then.
And I think from the point of that, I mean, the more I dive into medicine, the more remarkable is it that you can even be alive, because it can go wrong. So many things that can, cause issues. Today we talked about the endothelial glycocalyx, but obviously the endothelial glycocalyx is something that works together with everything else in the body. And it makes one, it has one specific, well, it doesn't have one, it has specific functions, within the functions of everything else that happens in the body, right? So, that's just true. And hopefully by manipulating single systems, we can help patients. But in reality, it really is trying to figure out how it works together as a whole.
Yeah.
That, I mean, I can lay awake at night and try to think about these things, but it gives me a headache. So.
Right. Not enough sleep. And then you're back at the beginning.
Exactly.
Ruining your glycocalyx. Yes. Good. All right. Any last thoughts?
Oh.
That's pretty good. All right. Well, thank you so much for being here with us. And We love that you've joined us today to discuss how to make our whole world medicine. We rise or fall together, and we're committed to sharing knowledge and insight that empower us and our communities towards greater health. Thank you for listening to the Nervata podcast for complex health, and we'll see you next time.